In today's video, we're going to discuss about the qualitative analysis of the known carbohydrate starch. So we know that starch is present in vegetables like potato, rice, so on and so forth. So let us discuss about starch today. So we know very well that the practicals are of two types. One is the qualitative analysis and the second one is the quantitative analysis. In qualitative analysis, we find out whether a particular compound is present or absent. But whereas in quantitative analysis, we know that a particular compound is present, but we don't know how much is present. So qualitative analysis of carbohydrates. So in this class we will discuss about the tests that are used to analyze the carbohydrate starch, how to perform the experiment, how to note the observation and how to give the inference for the results obtained. So these are the list of tests used for qualitative analysis of carbohydrates. We have discussed this previously in analysis of glucose, fructose, arabinose, maltose and so on. So to find out if a particular sugar is reducing or not, we perform the set of first 8 experiments, and then if we find out in the due course that it is not a reducing sugar, then we go to the 10th step, that is the hydrolysis step. So the list of tests are a solubility test, Malicious test, iodine test, Barfoot's test, Benedict's test, Felling's test, Bayal's test, Selivanov's test, phenylhydrazine test, and then finally hydrolysis test if the carbohydrate is a non-reducing sugar. So first test is the solubility test. So solubility of the substance is noted in the water, and starch we know it is a polysaccharide. So we will observe that it is insoluble in water and we will infer that presence of polysaccharides. And next test is malicious test. So malicious test is performed to identify the presence of carbohydrates, and since starch is a carbohydrate, we get a violet ring at the junction of the liquids. And next is the iodine test. So starch being a polysaccharide will interact with iodine and produce a deep blue color that shows the presence of starch. And next test is the Barfoot's test, which is performed to identify a monosaccharide. So since starch is a polysaccharide, we observe a negative reaction for Barfoot's test. And next is the Benedict's test. So starch is a polysaccharide and it doesn't have its reducing ends free, so we get a negative reaction for Benedict's. And same is the case with Felling's. Since starch doesn't have its reducing ends free, we observe a negative reaction for Felling's. And it also is not a pentose, so we don't get a positive test for Bayal's. And starch also doesn't have a keto group, so we get a negative reaction with Selivanov's test. So hydrolysis step. So this is very important. Since all the tests prove that starch is not a reducing sugar, we carry out with the hydrolysis step. So to 5 ml of the test solution, added 0.5 ml of concentrated HCl, and then place the test tubes in boiling water bath for 10 minutes, and then check the hydrolysis by adding a drop of iodine in a test tube and a drop of the hydrolyzed solution. If blue color persists, then hydrolysis is not complete. If the color of the iodine does not change, then hydrolysis is complete. Now neutralize the test solution with sodium carbonate and add sodium carbonate little by little till the effervescence subsides. Also check for the complete hydrolysis with Benedict's solution and Felling's solution. After complete hydrolysis, check for the ozone formation with phenylhydrazine. After hydrolysis is done, what are all the tests that have to be performed after hydrolysis? So number 1, iodine test. Number 2, Benedict's test. Number 3, Felling's test. Number 4, Barfoot's test. And number 5, phenylhydrazine test. So iodine test is performed, and since the solution is hydrolyzed, no characteristic reaction is obtained. So that shows that hydrolysis has occurred. Benedict's test. So when we perform Benedict's test, the color changes from blue to green to yellow to orange to red depending upon the degree of hydrolysis, and that confirms the presence of reduced sugar in the hydrolyzed solution. Next is the Felling's test. So Felling's test is also performed to find out whether a particular solution has reducing sugar or not. So after hydrolysis, starch gives positive for Felling's test. That shows that the hydrolyzed solution contains reducing sugars. And next test is the Barfoot's test. So if the starch is completely hydrolyzed, then it gets converted into glucose finally. So Barfoot's test will give positive for the hydrolyzed starch solution. So here we have to remember one thing, that starch, if partially hydrolyzed, might contain maltose, a disaccharide, and if it is completely hydrolyzed, then it will contain the monosaccharide glucose. So it's a very important step that we have to remember. And next is the phenylhydrazine test. If the substance is completely hydrolyzed, we get yellow needle shaped crystals, that is glucose ozones, within 10 minutes. So that confirms the presence of glucose in the hydrolyzed solution, which proves that starch is made up of glucose as its basic moiety. So that is an important lesson we have to carry today. Starch is made up of the glucose monomeric units. Partially hydrolyzed starch solution will have maltose, and completely hydrolyzed starch solution will have glucose units in it. So from this we will report that from the experiment, it was observed and reported that starch is a non-reducing carbohydrate made up of monosaccharides, that is glucose, because we obtain glucose zone.